Hi everyone. We have finished with the chapter Biomolecules, and just when it is fresh in our mind, we will begin with plant physiology, specifically the chapter Photosynthesis. Okay, it will be easier for you to understand all the pathways and all the enzymes because biomolecules are fresh in your mind. Fine, so let us begin. Now, we require energy to survive. Just in the last class, we discussed the living state, in which I said that metabolism is required to generate energy. Where does metabolism generate this energy from? So it gets its energy from food we eat, right? So we require this energy which we are getting from metabolism, and metabolism of what? So metabolism of food, right? So we can get this energy, as I said, by eating food that is from our diet or by making our own food. There can be two options. The first type of living organism are called heterotroph. That is, they get their energy and they get their food from other living organism. Okay, they are dependent on other living organism for food. And the autotrophs are the ones which can make their own food. Clear? Examples of heterotrophs are animals. Okay, all the animals that you see either eat plants or insects or other animals, right? So they are dependent on other living organism for their food. Same is the case with humans. We are also dependent on um, vegetables and fruits in case of vegetarian diet, and in case of non-vegetarian diet, we are also dependent on eggs and meat and all from other animals, right? Fungus is also dependent on other living organism. It can grow either on dead or decaying as well as live plants. Okay, you must have seen in forest and all a lot of fungus grows around, right? So it can live on dead and decaying matter as well as it can live on living matter also. And finally, you have bacteria. Bacteria, they also survive on other organism, okay, or organic molecules from other organism. Then comes autotrophs. The autotrophs include plants. All the plants that you see, especially the green part of the plants, they are all autotrophic. Then you have algae, which is nothing but unicellular plants, okay. They were previously considered under plant kingdom, but now they are considered under protista. And finally, you have some bacteria which are also autotrophic. Okay, example is cyanobacteria, which was also known as blue-green algae. Okay, it is a misnomer. That is, they were falsely called blue-green algae. They are actually bacteria. Clear? So all of these are autotrophs. Now the question may arise: how do autotrophs synthesize their own food? How are they making their own food? So they are doing it with help of photosynthesis. Okay, and what is photosynthesis? It is a physico-chemical process by which solar energy is converted into chemical energy. Okay, so whatever solar energy, the energy from the sun, it is converted, it is used to make bonds between different molecules and different atoms. So it is used to make chemical energy, which is nothing but your sugar. Fine. So with this in mind and with this background, let us start. So what do we know till now? You must have um, done some experiments or heard about it somewhere, or maybe even on the television, in those science programs, people do such kind of experiments to prove something, right? So what we know is following components are required by plants to carry out photosynthesis. Let us discuss all these components one by one. We know that the green part of the plant, okay, the chlorophyll, which is a green pigment, is necessary for photosynthesis, right? So this here is the structure of chlorophyll. As you can see, this ring-like structure is called porphyrin ring. In middle of this porphyrin ring is magnesium ion, okay, and it will also have a long carbon chain which is shown here as C20. Okay, so there will be 20 carbon long chain. This here is the basic structure of a chlorophyll. So we know chlorophyll is required for photosynthesis. Second thing, we also know that carbon dioxide is required for photosynthesis. How do we know this? So I don't know if you have done, and if not, let us just discuss. There was a potassium hydroxide experiment. Basically, what you do is, as shown here, you take a flask, and in this flask, you insert a leaf. Okay, so now you have a leaf in a flask. Along with the leaf, you add potassium hydroxide, that is KOH crystals. Potassium hydroxide loves to absorb carbon dioxide. Okay, it will absorb all the carbon dioxide. So after entering this leaf inside the flask, you seal it from above. Okay, the flask is sealed. What will happen is all the carbon dioxide in the flask will be absorbed by this potassium hydroxide. So the leaf no longer has free carbon dioxide to work with. Okay, it does not have carbon dioxide to photosynthesize with. After a while, when you take this leaf out and you put iodine on it, I hope you remember we had done this iodine experiment in biomolecules, right? If you put iodine and if it turns blue-black, it means there is presence of starch. Here, what will happen is, because this leaf was not in contact with any amount of carbon dioxide, it was not able to do photosynthesis, which means the leaf will not show any starch deposition. The same thing is done here. As you can see, you can take a leaf, break it, and put it in water so that it does not dry. Half of the leaf you can keep it inside, half will be outside. In the jar, you fill it with potassium hydroxide. Again, the same thing. It will absorb all the carbon dioxide from the jar. After a while, when you take this leaf out and you process it and add iodine to it, you can see only the outside part, which was in contact with air, which was in contact with carbon dioxide, was able to synthesize starch or was able to synthesize carbohydrate. The green part, which was inside, was not able to do photosynthesis. So this proves that carbon dioxide is required for photosynthesis. Okay, it is absolutely essential. Last thing was light. This also, I hope you have done this experiment, very simple experiment. You take a leaf. On that leaf, this could be done even on a living plant. Okay, it does not have to be broken off. So on that leaf, you keep a black colored paper. Okay, so that the sunlight does not fall on that part. You leave it like that for a day, and after that you break that leaf, you put it in alcohol and water and you boil it. This boiling is done just to weaken the structure of the leaf, okay, so that iodine can be absorbed. After doing this process, when you add iodine on it, the portion which did not have the black paper will turn blue black in color, showing that there is activity, there is starch which has been synthesized. And this starch came from where? It came from glucose, and this glucose was synthesized by photosynthesis. In the middle portion, which is green in color, because there was a black paper, the sunlight could not reach that part, and so that part did not perform any photosynthesis. So there will be no blue-black color. Clear? So these are the few simple experiments that you may have done or may not have done. If you haven't done, then I hope you have understood right now when we discussed it. Okay, so these are the simple experiments which show you that what all components are very, very important for the process of photosynthesis. To study how the scientists found this out, okay, so we will be studying the timeline of discovery of photosynthesis. How we discovered what this reaction was, what this process was. Okay, here again we will be discussing only the main scientists, also the scientists which are just mentioned in your textbook. Okay, mostly those and few others which are important. All of their study will not be discussed. So if you are interested, if you want to find out each and every kind of study, I suggest you go to this link. Okay, you can take You can pause and either note it down, or whenever you want, I can give you this presentation and you can go and visit this site for a whole timeline. Fine. Now let us start with whatever is important for us, and let us find out how the scientists discovered this process of photosynthesis. The first name that comes up in photosynthesis is of Joseph Priestley. This fellow, this scientist, he was living in 1770s, okay, and in 1774 he also discovered oxygen. So he has done work in case of botany also, as well as in terms of chemistry also. Fine. He did some experiments which proved that there is essential role of air in growth of green plants. He proved that there is something in the air which is essential for the growth of green plants. Okay, so let us discuss what he did, what the experiments were done. He took a bell jar. Okay, this is a bell jar, and he took a candle. He lit the candle and put it under the bell jar. What happened was, for some time, for a very brief moment, this candle was lit and suddenly it got extinguished. Of course, because there was no oxygen supply which was coming in. He did the same experiment with a mouse. He kept a live mouse inside a bell jar, and he saw that within few minutes the mouse was dead. From this, he said that there is something that the candle and the mouse are adding to the air, which is making it impossible to breathe in. Okay. Still, he does not know what component it is, what compound it is, but he is just saying that the candle and the mouse, they are releasing something in the air, which is not allowing them to live. Next, what he did was he took a small mint plant and he placed the mint plant along with a lit candle. He placed the bell jar upon this whole setup, and he saw that the candle stayed lit for a long time. He did the same kind of experiment with a mouse, and he saw that the mouse was also living for a long time. With this, he concluded that the plants restore to the air whatever breathing animals and candles remove. Okay, so he said plants are restoring something. Okay, they are restoring whatever the candles and the mouse are taking in. Clear? And later on, of course, we found out that this was nothing but oxygen. Right? So in photosynthesis, we all know oxygen is released. Right? So this oxygen is coming from no one else but the plants and the leaves itself. After Joseph Priestley came Jan Ingenhouse. Okay? Jan Ingenhouse. Now what he did was he said that sunlight is essential to the plant process. Okay, whatever the plant process was shown by Joseph Priestley, he said for the same process, sunlight is also essential. Okay, and it purifies the fouled air fouled by burning candles or breathing animals. Clear? So what he did was he did the same experiment. He took a candle, he took a mint uh, plant, he put a bell jar on it, and he kept it under light. He saw the candles stayed lit for 7 days. Okay, the candle stayed lit for 7 days. He took the same setup of candle, mint plant, and bell jar and kept it in the dark. In the dark, it did not last for even a few minutes. The reason is the plant cannot do photosynthesis in absence of sunlight. So, because there was no photosynthesis, there was no oxygen released, and so the candle extinguished. So, from this experiment, he said that sunlight is essential for the process which Joseph Priestley described. Second thing, when you observe, you can do this experiment if you want. Okay, just take some transparent glass or plastic bottle and put some plant or some what you can say twig in it. Okay, if you have mint or pudina, what you call or any such plant growing in your home, or if your mother has brought some bhaji, then just take one whole stem and put it in a glass bottle or a plastic bottle and fill it with water and look at it. Keep it in sunlight. Okay, you will see small bubbles like this forming. Okay, on the green parts of the plants. So these bubbles are nothing but oxygen. Here, as you can see, when you provide with sunlight, this leaf, okay, the green part, it is synthesizing oxygen, and this oxygen will displace water. As it is lighter than water, it will rise. Okay, and you can see it above here in the test tube. Okay, so he showed that it is only the green part of the plants that could release oxygen in presence of light. He saw some small bubbles like this on the green part of the plant and concluded that only the green part of the plants did the process and released oxygen. After him, after Jan Ingenhouse, came Julius von Sachs. Julius von Sachs, he provided evidence that there is production of glucose in plants. Okay, this scientist, he said that there is production of glucose in the plants. His studies later showed that the green substance in the plants, okay, what the Jan Ingenhouse has said, that the green part of the plants were synthesizing oxygen. So he showed that that green part, okay, that green substance was present in some special bodies. These special bodies are nothing but chloroplast, inside which there is chlorophyll present. Clear? So here you can see this is the whole cell, and inside the cell these are small chloroplasts, inside which the green photosynthetic pigment is present. Clear? So he was the one who showed that there are some special bodies inside which this green pigment is stored. He found that the green parts in plants is where glucose is made. He also found out that these are the parts where glucose is actually made, and it is usually stored as starch. He also showed that the glucose which is synthesized in these green parts is stored as starch. And this was the contribution of Julius von Sachs. So by the middle 19th century, okay, by the middle of 19th century, the empirical equation for photosynthesis was considered to be this. From all the scientists and all their conclusions, we come to this formula, that there is carbon dioxide which is required, so CO2. There is water which is required, of course, so H2O. There is some carbohydrate which Julius von Sachs said it is glucose. So there is this carbohydrate, or you can even call it starch, which is synthesized here. Okay, so CH2O, this is showing carbohydrate, and as shown by Joseph Priestley and Jan Ingenhouse, there is oxygen which is released. So this was the empirical equation. This was somewhat the equation of photosynthesis in middle 19th century. After that, there was a scientist called Cornelius Vanneel. Cornelius, he studied purple-green bacteria. As you can see here, can you see this is purple or violet colored bacteria, right? Actually, the bacteria are present in such a high number that you find the color only of that place turning into purple. Okay, they are not this big in size, of course, but they are higher in number. So you find the whole place turning purple or green in color, as you can see here. So he studied these purple and green bacteria. Now he showed that photosynthesis is a light dependent reaction. This much we already know, in which hydrogen and electron from a suitable compound reduce the carbon dioxide to form carbohydrates. So he was the first one to say that there is hydrogen and electron. Okay, somebody is donating this hydrogen and electron to reduce the carbon dioxide. Okay, carbon dioxide, only when it gets reduced it will turn into carbohydrates. So this he expressed by 2H2A. So this H2A, in this H2 is the hydrogen and electron donor. Okay, plus carbon dioxide will give you what? So it will give you this carbohydrate, which is nothing but reduced carbon dioxide plus water and plus 2A. Clear? So this was the reaction he gave. In case of purple and green bacteria, okay, this H2A is actually H2S, hydrogen sulfide. If you know hydrogen sulfide, it tastes like, it smells like rotten fish and rotten eggs. Okay, it smells very bad. So this H2S was actually being used by this green and purple sulfur bacteria. They used hydrogen sulfide as hydrogen and electron donor. Clear? So here H2A will be H2S in case of green and purple sulfur bacteria, and in case of plants, okay, in case of algae and higher plants, this H2A is actually water. So CO2 plus H2O will give you what? It will give you carbohydrate. Clear? And that is how the whole equation for photosynthesis was derived. Hence, so from this, what do you know? So if you use H2S, okay, H2S plus carbon dioxide, they saw that you were getting sulfur, which was getting released, okay, sulfur plus carbohydrate. There was no oxygen which was evolved by green and purple sulfur bacteria. This proved that oxygen was not coming from carbon dioxide. Okay, the oxygen which was released by the plants was not coming from carbon dioxide, but rather it was coming from water. This again was proved by Rubin and Kamen. Okay, these were the two scientists who proved it. They did what is known as radioisotope technique. I hope you have I don't know, studied radioactivity also in chemistry as well as physics. Radioactivity, you can make any, you can have any compound which can have radioactive isotope. Here we used two, what you can say, two different molecules which had oxygen radioactive isotope. So oxygen was taken was 18, O18, which is radioactive. Okay, so it can be traced. In experiment number 1, what was done was they took normal carbon dioxide, but the water was radioactive. The water given to the plants was radioactive. There was light which was given to the plants, and after that they saw whether the glucose was radioactive or the oxygen was radioactive. To their surprise, the oxygen that was evolved was radioactive. This meant that it was from water that the oxygen was getting produced. Okay, so from water only this oxygen was produced. To be sure, they did another experiment. They took radioactive oxygen labeled carbon dioxide. They added normal water, gave light energy, and saw that the carbohydrate turned radioactive while the oxygen was not radioactive. This again proved the same point, that oxygen was not coming from the carbon dioxide, but rather it was coming from water. Clear? Fine. Then there was another scientist called Robert Hill. What Robert Hill did was he demonstrated, he first thing he did was he isolated chloroplast. Okay, so from a plant cell, he removed everything else and he got chloroplast separated. He took these chloroplasts in a solution, okay, like this, and he showed that even in presence of some, in absence of carbon dioxide, there was no carbohydrate produced. That is obvious that carbohydrate is required for carbohydrate production. But when he gave what you can say, this reducing equivalents, okay, artificial electron acceptors, ADP and NADP, and water, he saw that the chloroplast took this in and still produced oxygen, ATP and NADPH without production of carbon dioxide, okay, when in absence of carbon dioxide. So this proved that the isolated chloroplast, okay, in presence of an artificial electron acceptor, in presence of any artificial electron acceptor, something which will just accept the electrons, The chloroplast can evolve oxygen in absence of carbon dioxide. So even when there is no carbon dioxide, if you give the chloroplast ADP and NADP, it will still produce oxygen. Yeah. So here we prove the same point that from water we are getting oxygen, not from carbon dioxide. So concluding all of these years of research and all of these discoveries by the scientists, the final equation representing photosynthesis is 6CO2, which is carbon dioxide, plus 6H2O, water, in presence of sunlight and with help of chlorophyll will get converted into glucose, which is C6H12O6, plus 6 molecules of oxygen are released. Note that this is not a single reaction. Okay, you cannot complete this whole thing in a single reaction. It is a multi-step process, and this whole multi-step process is called photosynthesis. What is the importance of photosynthesis from whatever we studied till now? First thing, it is a primary source of all food on earth. Only because photosynthesis is present that the sunlight and the carbon dioxide are getting converted into glucose. This glucose is required by all living things for their survival, right? So it is providing food for every living organism on earth. Second thing, it is also providing with oxygen. So it is giving oxygen in releasing oxygen into the atmosphere, which is also required for respiration of this glucose. Okay, for food to be digested and to be absorbed, we require oxygen. To make it converted into energy, we require oxygen. Also, during this process, there is a release of carbon dioxide. This carbon dioxide is again used up by the plants to produce more and more glucose. So plants, they balance everything out, and they are able to do this because of photosynthesis. Here we end today's lecture. Tomorrow we will start with the next part of photosynthesis. Okay. Again, it will be somewhat like introduction, finding out what all players are there in case of photosynthesis, and we will see a little basic about photosystems, light harvesting complex, etc. Okay. See you tomorrow.